Hello, this is lecture 16, in which we will talk about protein folding in the ER. Now, in lecture 15, I said we were going to talk about three things: glycosylation of proteins, the process whereby we add carbohydrates to proteins, the formation of disulfide bonds, and folding and assembly of multi-subunit proteins. Now, we talked about the first two, but as that lecture unfolded, I felt like it was getting too long, and so I decided to break it up. So suffice it to say, we've covered this and we've covered this, and now in this lecture we're going to focus on this. So that's what this is, really just an extension of 15. It's just a separate lecture that will focus on protein folding. Now, you shouldn't be surprised to learn that when we talk about protein folding of proteins in the ER, it's going to involve chaperones, because we've seen over and over how important chaperones are in the protein folding process. We've seen chaperones in the beginning of the semester when we talked about protein folding in general. We saw both cytosolic and mitochondrial HSP70s and the role that they play in import and protein folding of mitochondrial matrix proteins. So it turns out in the ER, there's a protein that plays a really important role, a molecular chaperone, an HSP70 chaperone called BIP. So we're going to see what BIP does, and we'll see that BIP is going to play a really important role in this process in the ER. Now just remember that it's not surprising that we would use a molecular chaperone for protein folding, specifically an HSP70 molecular chaperone, since they work at the co-translational level. So here's BIP. Simple slide here. We've got our translocon, we've got our ribosome here, and we've got our protein moving through the translocon. So as that protein emerges and gets into the lumen of the ER, these hydrophobic patches, these exposed hydrophobic patches, are bound by this protein called BIP. That's exactly what that chaperones do. Remember, they're going to recognize exposed hydrophobic amino acids, because in a fully folded protein, those hydrophobic amino acids, as we know, should be buried within the core. Well, here, this protein isn't folded yet. It's just emerging through the translocon. And so those hydrophobic patches are exposed and they're bound by BIP. As the protein begins to fold, then, those hydrophobic patches become buried on the inside where they should be, and that's going to cause the BIP to come off. This is exactly the way chaperones work. Now, it involves ATP and all, as we've seen earlier, but this is the way chaperones work to facilitate protein folding. So here's the bottom line. BIP is going to play a really important role in facilitating protein folding. It's essentially going to keep the protein unfolded until essentially the whole protein emerges so that it can fold properly. Exactly what we saw for HSP70 chaperones in the cytosol. Now, remember I told you in lecture 15 to keep an eye on these three glucoses, right? These three glucoses that are added to proteins that are moving into the lumen of the ER. We said at the time that one of their roles was to signify that that's a fully formed precursor, so that oligosaccharyl transferase will just transfer a fully formed precursor to a protein. We also know, and I think we'll see it on the next slide, that those terminal glucose residues are removed by glucosidase 1 and glucosidase 2, which are consistent with their role as indicators of completeness when it comes to the precursor. You can see that right here. They're going to be removed by glucosidase 1 and then glucosidase 2. That event is taking place in the ER. We'll have another minor modification in the ER, but then, as we saw in lecture 15, the vast modification that occurs takes place in the Golgi apparatus. So before we figure out, as we saw, those three glucoses are going to be removed, that's a signal that as we've said over and over now, that the preformed precursor is ready to be added to the protein. Once that protein is in the lumen of the ER, it's going to fold spontaneously. BIP is going to help it. That's what chaperones do. But protein folding is spontaneous, so there's really not much to talk about in terms of protein folding in the ER. Really what we need to focus on is what happens if protein folding doesn't occur properly. Because if you think back to the beginning of the semester when we talked about protein folding, what we found was that if a protein doesn't fold properly, the cell's going to invest some energy to try to properly fold that protein. Okay, and we're going to see that again here. And if protein folding doesn't occur properly, eventually the cell gets rid of the protein. Well, let's ask the same question in the ER. As that protein emerges through the translocon, BIP binds those hydrophobic pockets, as we've already seen, patches rather, keeps the protein from folding. Once the protein is fully made, then it can fold. Okay. Let's assume it doesn't fold properly. Well, here's what's going to happen to that protein if it doesn't fold properly. There's an enzyme in the ER called UDP glucose glycoprotein glycosyl transferase. So let's see what that protein does. It's going to be, we'll call it UGGT hereafter just because its full name is a mouthful. But look at what it is. It's a glycosyl transferase, so that means it's going to transfer a sugar. Specifically, the sugar it's going to transfer is glucose. It's going to get that glucose from UDP, and it's going to transfer it over to a glycoprotein. In other words, it's going to transfer it to a protein that already has carbohydrates on it. This is a really important protein. It turns out that it's embryonic lethal. So what that means is that so what can happen in biology a lot of times, if you want to know how important a protein is, you knock out the gene that encodes that protein and you ask what happens to an organism that doesn't have that protein. And it turns out if you knock out the gene that encodes this UGGT protein, that it's embryonic lethal. That means that mice die in utero. If knocking out a protein is embryonic lethal, it tells you that that is a really, really important protein. In other words, it's not as if the mouse is born and it's just got some abnormality. It can't even be born. And so here's what this UGGT does. You've got a misfolded protein right here. That misfolded protein already has its carbohydrate tree on it. And remember, this is UGGT. So what it's going to do is it's going to take a glucose that is attached to UDP and it's going to transfer it over to the carbohydrate tree that already exists. So the dark brown here is glucose, the light brown is mannose. Now, why would it want to do that? Why would it want to take those glucoses that it took off and put them back on? Because that's what's happening. That's why I told you these glucoses are so important. Not only do they serve as a signal that the preformed precursor is complete, they're also going to play a role in protein folding. And this is the way they play a role. Once that glucose gets added back, it gives a protein called Calnexin a high affinity for that protein. Now, what is Calnexin? Calnexin is a lectin. And lectins are proteins that have a high affinity for other proteins with carbohydrates on them. So they're essentially carbohydrate binding proteins. There are a couple in the ER that are important, Calnexin and calreticulin. This slide really focuses on Calnexin. So notice that Calnexin is a transmembrane protein with its active site in the ER lumen. It exists as a complex with ERP57. Now ERP57 is going to allow disulfide bond formation or rearrangement, and it's going to allow it's going to help facilitate protein unfolding. So here's what happens. Our misfolded protein gets that glucose added back on it. It then, because it has that glucose added back on it, has a high affinity for Calnexin. So Calnexin holds on to it, and Calnexin is next to ERP57, so ERP57 starts breaking and rearranging those disulfide bonds, and unfolding that protein. Now, what then happens is after the protein is released by ER-P57, the glucose is clipped off again. Now the protein can try to refold, and so the protein is going to refold. If the protein is properly folded, it will be exported from the cell. Remember mannosidase 1, before it leaves the ER, not only do we take off those 3 glucoses, but we take off 1 mannose. So here we're going to take off that 1 mannose before it gets exported to the Golgi apparatus. So if that's what happens, all is well and good. That protein didn't fold properly the first time, but that's okay. It had a glucose added back to it. That caused it to be bound to Calnexin. ER-P57 unfolded it. It refolded properly the second time. Now it's incorporated into a vesicle and taken to the Golgi apparatus. But what if it doesn't refold properly the second time? Well, we're just going to repeat the process. We're going to add a glucose to it again and go through that whole sequence of events again until hopefully it gets properly folded. Now, all the way at the end of this lecture, we'll figure out what happens if it never gets properly folded. Let's not worry about that now. I'm not going to spend any time on this slide because it's exactly the same as the previous slide. If you take a look at this, you'll see that it's describing exactly the same sequence of events. Now what we want to do, though, is ask ourselves, in general, not if you're looking at a specific protein, but in general, if there's a protein folding problem in the ER. This is a really cool thing we're going to discuss here. Again, it shows you just how incredible the cell is and how the cell works so hard to make sure everything happens as it's supposed to happen, including protein folding. So let's just say, for example, that we've got a protein folding problem for whatever reason. Maybe the protein has a mutation in it. Maybe there's an amino acid substitution, so the protein's not folding properly. Maybe environmental conditions are making it difficult for protein folding. It doesn't really matter. Let's just define it as a protein folding problem. Well, it turns out that in the ER, there are 3 proteins that have sensors for the presence of misfolded proteins. And these proteins are called ATF6, PERK, and IRE1. Okay? So we're going to take a look at these one at a time. We'll start with ATF6 and move to the left. But what I want you to see at the outset is that all 3 of these have sensors for misfolded proteins. So in other words, they're all transmembrane proteins, as you can see. This is the ER membrane. This is the luminal side. This is the cytosolic side. Whoops. Here's the sensor portion right here. That indicates the sensor, and the sensor. Now, what is the sensor looking for? Well, it's looking for the typical characteristic of a misfolded protein, and that would be exposed hydrophobic amino acids. Now, let's say that you have exposed hydrophobic amino acids. In other words, we don't have proper folding of a protein, and the sensor for ATF6 recognizes that. In other words, that sensor is going to have a high affinity. What's going to happen then is ATF6 is going to be incorporated into a vesicle that goes to the Golgi apparatus. Once it gets to the Golgi apparatus, it's going to encounter a protease. So here's your protease. That protease is going to clip ATF6 in the Golgi apparatus. Now, where's it going to clip it? It's going to clip it right here. Now, remember, this is a transmembrane protein. So the portion that's in the cytosol is going to remain in the cytosol. So if ATF6 is incorporated into a vesicle that goes to the Golgi apparatus, once it gets to the Golgi apparatus, the domain that was in the cytosol remains in the cytosol. And so if we clip it at this spot right here, as we said, then we're going to liberate this whole portion. It's no longer going to be anchored to the membrane. Well, what is that going to do? It turns out, amazingly, that that domain is a transcription factor. That domain is going to go to the nucleus, and when it gets to the nucleus, what do you think it's going to do? Here's a gene. We'll just draw one cis regulatory sequence right here. This transcription factor is going to bind right here, and this gene is going to encode a chaperone. Now that's pretty remarkable if you think about it. It's as if the ER says to the nucleus, I need help. I've got a protein folding problem. I have proteins that are not properly folded. I need more chaperones to help with the protein folding process. So I'm going to send you a transcription factor. I need you, nucleus, to use that transcription factor to upregulate the expression of genes that encode chaperones so we can make more chaperones and hopefully help with this folding process. Now, is that cool or what? That's the ATF6 mechanism. Let's erase that. Whoops. And let's look at the second mechanism. The second mechanism involves a protein called PERK. Now, here's what's going to happen with PERK. PERK is going to be involved in, well, before we talk about PERK, let me just ask one other question. If you're the ER and you have a protein folding problem, we've already seen with ATF6 that one thing that would help with that protein folding problem is more chaperones. But can you think of something else that might help with that protein folding problem? Something else that the cell could do, doesn't have anything to do with chaperones, that might help? Well, if you've got a protein folding problem, the last thing you need to do is keep adding proteins to the ER. The more proteins you add to the ER, sounds like Winnie the Pooh here. The more proteins you add to the ER, the more concentrated the proteins become, the higher the density. The higher the density, the more difficult it is to fold. And so really, it's again, in this case, it's as if the cell says, it's as if the ER says, well, let's not bring any more proteins to the ER to make things more difficult than they already are. So what you can see is that PERK is going to be involved in reduction of proteins entering the ER. Okay, so let's look at what happens here. It turns out that when PERK senses misfolded proteins, that we're going to activate a kinase domain. Okay, and that kinase domain, as we'll see on the next slide, is then going to be responsible for phosphorylating something that reduces protein entry into the ER. So let's go to the next slide and see how that works. Now, in order to kind of see how this works, we've got to go back and remind ourselves of what's going on when it comes to translation. So remember, if we look at this mRNA right here, that AUG is part of the Kozak sequence. And remember what we said is on the 5' end of that mRNA, we're going to assemble a translation initiation complex. That translation initiation complex is then going, including the small ribosomal subunit, is going to scan until it reaches the Kozak sequence. And then we'll finish assembling the ribosome and translation can begin. Now, in order for that to happen, you need this protein called EIF2. EIF2 is a G protein. You can see it right here. So you got EIF2, you've got your tRNA that has methionine attached to it, and you have your ribosome, and then you have some other proteins here. We don't need to worry so much about what those other proteins are, the EIF1A and the EIF3. The key one for this is EIF2. And notice that EIF2 here has GTP bound to it, so it's an active G protein at this point. So you need that EIF2 to form that translation initiation complex. Okay, that's the thing to remember. You need that to form the translation initiation complex. Now, like all G proteins, EIF2 will become inactive when the GTP is converted to GDP. So let's say that you've got an inactive EIF2. Well, how are you going to activate EIF2? Same way you activate every other G protein. You need a GEF. So here's the GEF, guanine nucleotide exchange factor. That's the GEF for EIF 2. So here it is. Okay, it's going to bind to EIF 2. The GDP is going to be removed and replaced by GTP. Now you have your active EIF2. Now that's what happens under normal conditions. Let's go down to the bottom and see what happens. There is a protein kinase that can phosphorylate EIF2. If that protein kinase phosphorylates EIF2, then EIF2, when it's in its inactive state, because it has GDP bound to it, will bind to the guanine exchange factor, the GEF, EIF2B is the GEF, but the EIF2B, the guanine exchange factor, can't do its job. So what happens is this complex just kind of sits here like this. Now what that means is that if you can't replace the GDP, you cannot get new EIF2 GTP. If you can't get new EIF2 GTP, then you can't form this complex. If you can't form that complex, then you can't get translation. So you're going to get no translation. So let's summarize it like this. If you have anything that phosphorylates EIF2, you're going to shut down translation. You're going to shut down translation because you cannot form that translation initiation complex that assembles on the 5' end of the mRNA. Now, as we just saw on the previous slide, if you phosphorylate that EIF2, you can't get translation. And we know that we're going to phosphorylate EIF2 if PERK is activated, and PERK is going to be activated if the PERK sensor detects misfolded proteins in the ER. And that all makes perfect sense. You might look at that and say, okay, that's great. We're going to decrease the load of proteins going to the ER. That ought to make it a little bit easier for the protein folding to take place in the ER. But there's only one problem with that. If you can't translate proteins, then how is the ATF6 pathway going to do you any good? In other words, let's go back and think about the ATF6 pathway. ATF6 is going to recognize or sense the presence of misfolded proteins. It's going to move over to the Golgi apparatus, will generate that transcription factor. That transcription factor will go to the nucleus, where we will transcribe genes that encode chaperones. Well, that's great. We've got the mRNA, but we don't need the mRNA. We need the protein. So you would have to translate that mRNA. But we just said that this mechanism is going to shut down translation of mRNA. So that seems to be counterproductive. They seem to be working against each other. Well, again, this is remarkable because here's what happens. If you take a look at the left, this shows what happens under most conditions. In other words, this shows that we've got EIF4G, EIF4E bound to the poly A binding protein, the polysome as we described earlier. Now what's not shown here is EIF2 is going to be part of this whole complex as well. But notice that this complex begins at the 5' cap and is going to move. It's going to move over here where we're going to have the Kozak sequence. Okay, so EIF2 is part of this. Now EIF2 is not shown in this slide because this slide is not really, this diagram was not put together to illustrate what we're trying to illustrate, but you can use it for that anyway because you'll see why in just a second. But we've assembled, if we can't assemble that, then we can't get translation, right? That's what we just established. But here's the thing. Some mRNAs contain what's called an IRES, and IRES stands for internal ribosome entry site. IRES. It turns out that with an IRES, you don't assemble this translation initiation complex at the 5' end. So the process that we described of assembling this translation initiation complex at the 5' end and then scanning, looking for the Kozak sequence is not the way this works. Rather, if an mRNA has an IRES, then the ribosome binds internally and you bypass all that. That's why it's called an internal ribosomal entry site. And you might be thinking, so what? Well, here's the so what part. Chaperone mRNAs have IRESs. Now, there are other mRNAs that have IRESs as well, but chaperone mRNAs have IRESs. So think about the implications of that. What that means is that ATF6 can do its job. We can upregulate the transcription of those genes. We can make the mRNA, and we can translate that mRNA and make more chaperones, even though EIF2 has been phosphorylated and we can't translate a bunch of other mRNAs to make a bunch of other proteins. So remarkably, the cell is able to maintain a high concentration of chaperones, even though it's really shut down the translation of other mRNAs as it tries to deal with this protein folding problem. Now we're going to look at the third response. This is called the unfolded protein response, and the nomenclature gets a little bit messy here. ATF6 and PERK are also part of the unfolded protein response. In other words, it's the cell's response to the presence of unfolded proteins. But the IRE1 pathway was discovered first, and it was called the unfolded protein response. But it turns out IRE1 is just one specific type of response. Okay, another way to say that is the unfolded protein response is an umbrella term, and underneath that you have IRE1, PERK, and ATF6. Nevertheless, sometimes the IRE1 pathway still might be referred to as the unfolded protein response. Now let's take a look at what goes on here. We've got a misfolded protein. That misfolded protein, well, I started to go over here. We're gonna skip that. We're gonna go on this direction. We'll get that other one in just a second. So here's your misfolded protein. Your misfolded protein is gonna bind to IRE1. Now, when the misfolded protein binds to IRE1, it causes IRE1 to phosphorylate itself. So it has a kinase domain, and you'll get what's called autophosphorylation. That phosphorylation event then activates a ribonuclease domain. So a ribonuclease is a protein that's going to clip nucleic acids. Here's a pre-mRNA. This pre-mRNA has not been spliced. The ribonuclease is essentially going to remove this intron and splice this exon and this exon together. Now we've got a fully spliced mRNA. Now before we go any further, let's stop there because this is bizarre, right? We know how splicing works. We know that splicing involves a spliceosome. We know that splicing takes place in the nucleus, and we know that mRNA are not exported unless they're export ready. And to be export ready means that splicing has occurred. Well, this breaks all those rules. It turns out that the mechanism for splicing here is well understood. I used to have a slide in here, but there are multiple aspects to it. So I kind of took it out because I don't think we really need to go into all the nuts and bolts of how that splicing event occurs. Suffice it to say that what you've got is you've got this pre-mRNA that's sitting out in the cytosol. That pre-mRNA is not going to get translated because it hasn't been spliced. It's only going to get translated once it's spliced. Well, when does it get spliced? It gets spliced when the ribonuclease domain of IRE1 gets activated. That domain gets activated when we've got protein misfolding. Now the question becomes, once that mRNA gets translated, what does it produce? Well, it produces a transcription factor. That transcription factor then moves over to the nucleus where it upregulates the transcription of genes that encode chaperones. We make our chaperone mRNA. Our chaperone mRNA gets exported. It gets translated. Okay, the chaperone is moving through the translocon, and we can aid in the protein folding process. So this is a case. Well, let me back up. We've talked about how we regulate transcription factors. We've said that transcription factors are typically not regulated at the transcriptional level. This one is not regulated at the transcriptional level either, but it's totally different than any of those mechanisms that we saw before. It's actually regulated at the translational level. In other words, it's the rate at which we translate that mRNA. So if you think about what's going on with this IRE1 pathway, essentially what you have here is another mechanism for creating chaperones. So IRE1 and ATF6 are both devoted to increasing the number of chaperones that function in the ER to help with the protein folding process. Now they go about it in different ways, as we've seen. ATF6 has a pre-existing transcription factor that just gets released from the Golgi membrane, whereas in this case, we've got an unspliced mRNA that can only get translated once it's spliced. So you got IRE1 on the one hand, ATF6 on the other hand, responsible for making more chaperones, and you've got PERK, which is responsible for reducing the load of proteins in the ER. Now, if that doesn't tell you how important protein folding is, nothing does. Because what that tells you is that the cell is going to, pull out all the stops to try to make sure protein folding occurs properly. Now, superimpose that on what we saw with Calnexin and that whole system, how much effort the cell puts into trying to refold proteins that haven't properly folded. Now, before we leave this IRE1, we need to do one other thing because it's another remarkable part of this story. Now let's go over here and look at BIP. Remember, BIP is going to play a role in protein folding, right? Well, it turns out that IRE1 exists in the membrane, the ER membrane, as a monomer. It remains in its monomeric form under normal conditions, and it remains in its monomeric form because it has a binding site for BIP. Now, if BIP is bound to IRE1, what does that tell you about BIP? It tells you that BIP is not busy folding proteins. In other words, BIP, the chaperone involved in protein folding, isn't occupied with protein folding. Rather, it's bound to IRE1. That keeps IRE1 in its monomeric form. Now, let's say you have a protein folding problem. That protein folding problem is going to cause BIP to be released from IRE1, move to the lumen of the ER where it's engaged in a protein fold in efforts to properly fold proteins. Well, now that IRE1 is not bound to BIP, it's able to dimerize. The dimerization of IRE1 is what's responsible for the activation of the kinase domain. This is seen really nicely in this slide, not so well over here. We can see that it's dimerized right here, but you can't tell from this diagram exactly how it got dimerized. Well, this is how it got dimerized. It dimerized because became activated by phosphorylation, and then the ribonuclease domain was activated. But that dimerization event occurred because BIP is no longer bound to it. BIP is busy trying to fold proteins. Now, one thing I want to say, this is called a stressed cell. This is a stress response. The stress in this particular case, though, is protein folding, so don't get bogged down with that. In stressed cells, BIP is occupied with unfolded proteins. So stress is used in this slide kind of almost synonymously with unfolded proteins. So that's how BIP and the IRE1 pathway are linked. Now, let's say that all efforts to fold proteins in the ER fail. What's the cell going to do? Well, we know what happens if a cell can't fold proteins in the cytosol, right? We add a ubiquitin tag to it, we send it over to the proteasome where it gets degraded. And we do that because if those proteins were to hang around, they would aggregate, and that would be lethal to the cell. So what's the ER to do? Well, the ER could just say, well, I'm going to take this protein and move it on to the Golgi apparatus and let the Golgi worry about it. But the ER doesn't do that. The ER deals with these misfolded proteins. It could deal with the misfolded proteins in a couple of other ways. It could destroy them within the ER. If you're going to destroy it within the ER, then you have to have some sort of mechanism for tagging and destroying the protein, or you have to import the cytosolic mechanism that we've already described into the ER. Alternatively, you could actually take your misfolded protein and move it from the ER out into the cytosol and take advantage of the system that's already there. That's exactly what the cell does. So let's take a look at what's going to happen here. Here's a misfolded protein. Now notice it's shown with chaperone. That chaperone is trying to fold it. It's bound to a lectin, Calnexin or calreticulin. That's going to try to help with the protein folding process. But assuming that all efforts to fold this protein have failed, then what happens is it undergoes what is called retrotranslocation. And retro, of course, means backwards. So now we're going to translocate this protein from the ER lumen out into the cytosol. So here's your protein translocator complex. I don't think we know much about that complex. We just know that it exists and proteins can be moved in a retro direction. Now here's the amazing thing. Associated with the protein translocator complex is an E3 ubiquitin ligase. So what's that E3 ubiquitin ligase going to do? Well, remember what E3 does. E3 transfers ubiquitin from E2 over to the target protein. So this doesn't show this, but you would have to have an E2 here as well. But E3 is actually part of the translocator complex. So as that protein is being retrotranslocated out to the cytosol, a couple of different things are happening. It's being, well, the main thing that's happening is that ubiquitin is being added to it. And we're going to add ubiquitin just like we did before. That E3 ligase is going to transfer it over to lysine residues. You've also got this hexameric unfoldase, which is going to help to unfold the protein as it moves through that translocon. I mean the protein translocator. And you have a glycanase, which is a protease that's going to clip off the sugars. So when all is said and done, what do you have? You have a protein that has been deglycosylated, unfolded, and polyubiquitinated. It can then bind to the proteasome like any other protein, be degraded in the proteasome. This is the same proteasome that we saw in the cytosol, and there's our small peptides. So this protein that would not fold properly in the lumen of the ER has undergone retrotranslocation and degradation in the cytosol with via the proteasome. So pretty remarkable. That's how the cell is going to deal with protein folding. Specifically, that's how the ER deals with protein folding.